Chapter 13, Segment 3 starts with discussing we can store energy for future needs. Our most immediate source of energy are complex carbohydrates that are broken down to simpler sugars for us to use. These include glucose, and then glucose can be further broken down into glycogen, which is stored as a reserve fuel in several locations. The process by which this occurs is glycogenesis. Insulin is a hormone that is released by beta cells in the isolates of Langerhans that lowers blood glucose and promotes glycogenesis. Glucagon is then the hormone that's released by alpha cells in the isolate as opposed to the beta cells for insulin of Langerhans that increases blood glucose and it mediates the conversion of glycogen back into glucose. Glycogenolysis is then this process that converts glycogen back into glucose. Lipids are large molecules that consist of fatty acids and glycerol that are insoluble in water. They mostly make up adipose tissue, and under prolonged food deprivation, this fat can be converted into glucose and ketones. Ketones are just a metabolic fuel source in general. Glucogenesis is this process that converts body fats and proteins to create glucose. Next section is titled Insulin is Crucial for Regulation of Body Metabolism. The important role of insulin is mainly enabling us to use our body to use glucose. Glucose transporters are the molecules that conduct glucose molecules through the external membrane of a cell for use inside that must interact with insulin to function. There are 3 different sequential mechanisms that stimulate insulin release, the first being the selephic phase, second being the digestive phase, and the third being the absorptive phase. Glucodetectors are then cells that detect and inform the nervous system about levels of circulating glucose. Insulin converts glucose into glycogen, which is then stored in the liver and muscles. The vagus nerve is the cranial nerve 10, which regulates the viscera and transmits signals from the viscera to the brain. Glucodetectors then in the liver travel via this to the nucleus of the solitary tract, or the NST, which is just a brainstem nucleus. This system informs the brain about circulating glucose levels and contributes to hunger. Efferent fibers carry signal from the brain via the vagus nerve to the pancreas, where it modulates the insulin released from the pancreas. One of the diseases associated with insulin dysfunction is diabetes mellitus, which is excessive glucose in the urine caused by the failure of insulin to induce glucose absorption by the body. This can either be type 1 or type 2, which is a consequence of reduced sensitivity to insulin, which is usually associated with obesity. The next section is titled Despite their importance, neither insulin nor glucose is the sole signal for hunger or satiety. Hunger is the internal state of an animal seeking food. Insulin can provide this signal satiated to the brain, where low insulin makes you hungry, and when you increase insulin, you're not hungry. Glucose levels also are involved in this appetite signal, so it's inferred that the brain integrates many different signals to control appetite. The next section is the hypothalamus coordinates multiple systems that control hunger. Hypothalamus is critically important in the regulation of metabolic rate, food intake, and body weight. The VMH, or the ventromedial hypothalamus, is very important within eating behavior. It's usually known as the satiety center because when there's lesions in it, it creates excessive eating, which is called hyperphagia. Whereas the lateral hypothalamus is usually associated with the hunger center because lesions in it cause aphasia, which is a refusal to eat. But to control hunger, it's more complicated than just these two center controls. The next section is titled Multiple Peripheral Signals Are Integrated by a Hypothalamic Appetite Network. The arcuate nucleus is an arc-shaped hypothalamus nucleus implicated in the appetite control, and it's governed by circulating levels of a variety of hormones, including peptides, leptin, ghrelin, and peptide PYY336, or for short PYY3-36. Leptin is mostly important leptin's a peptide hormone that's released by fat cells, then secreted into the bloodstream. The brain, in monitoring the levels of the circulating leptin, measure and regulate the body's energy reserves in the form of fat. So it has long-term effect on appetite, and it's in low levels even when fat reserves are high in obese people. So it's usually a hunger controller. The next section is ghrelin. Ghrelin is a powerful appetite stimulant. This is apparent through the levels rising after fasting, and they drop after a meal has been eaten. In opposition to this, PYY3-36 is an appetite suppressant. This is apparent by its low levels in blood prior to eating, but then it's increased after eating. Next section is titled The Archaic Appetite System. This mainly relies on two sets of archaic neurons with opposing effects. The first one being the POMC, or the pro-opiomelanocortin, which is a prohormone that can be cleaved to produce the melanocortins, which also participate in feeding control. The neurons that have receptors for these are the POMC neurons, which are involved in the hypothalamic control system. They mostly act on satiety neurons and inhibit appetite and increase metabolism. In opposition to these POMC neurons, there's MPY neurons, which mostly act as hunger neurons when activated, and they stimulate appetite directly, inhibiting POMC neurons and reduce metabolism to promote eating and gaining weight. Projections from the POMC neurons and the MPY neurons leave the archaea and make contact with neurons in other hypothalamic sites. This is how the archaic system modulates food intake. Both types of appetite neurons in the archaic system have leptin receptors, mostly because leptin affects them in opposite ways. Leptin activates the appetite-suppressing POMC neurons and inhibits the appetite-increasing MPY neurons to suppress hunger for the longer term. Ghrelin and PYY are 3-36 are a little bit faster because they provide signals from the stomach. They act mostly on the MPY neurons of the archaea, whereas ghrelin stimulates it to increase appetite, and PYY 3-36 inhibits it to reduce appetite. The next section is second-ordered hypothalamic neurons integrate appetite signals. The archaic projections then go to the hypothalamus. The two different types of neurons are the orexigenic neurons, which promote feeding behavior, whereas the anorexigenic inhibit feeding behavior. The paraventricular nucleus, or the PVN, is implicated in the release of oxytocin and vasopressin and control of feeding and other behaviors. One set of projections from the PMC terminate here. So the alpha melanocyte stimulating hormone, or alpha MSH, binds to the melanocortin receptor, which is released by the PMC to the PVN. These melanocortins are just a category of endogenous opioid peptides from the POMC, and the type of receptors which are on the PVN are called melanocortin type 4 receptors, which are implicated in why alpha MSH activates PVN's appetite suppressant activity. In inhibiting anorexigenic PVN neurons, MPY releasing neurons promote increased appetite. By blocking the alpha MSH signal, AGRP counters appetite suppressing influence from the POMC neurons to increase feeding behavior. Orexin, or also known as hypocretins, are neuropeptides produced in the hypothalamus that are involved in switching between sleep states, in narcolepsy, and in the control of appetite. We usually increase feeding when they are in high levels. The next section is other systems also play a role in hunger and satiety. Appetite signals from the hypothalamus converge on the nucleus of the solitary tract, or the NST, in the brainstem, which receives and integrates appetite signals from a variety of sources. CCK, or cholecystokinin, is a peptide hormone released by the gut after ingestion of food when it's high in protein and or fat that also serves as a signaling molecule to the brain. It acts directly on receptors in the vagus nerve to inhibit appetite. The brain's reward system is also involved in food intake, and it usually mediates the pleasurable aspects of food. In addition, endocannabinoids usually influence appetite in feeding by stimulating feeding by affecting the mesolimbic dopamine reward system. It acts directly on the hypothalamic appetite mechanisms, and it also acts on POMC neurons, but instead of causing alpha MSH to stimulate hunger, to inhibit hunger, it releases B endorphins to stimulate hunger. Overall, the hypothalamic feeding control can also be influenced by inputs of higher brain centers. And that is Chapter 13, Segment 3.